This presentation was developed for Dalhousie University's medical entomology class, taught by Dr. Tatiana Rosalimo. It will discuss the Barmah forest virus found in Australia. The Barmah forest virus is the second most common arbovirus in Australia, following the Ross River virus, with 1,487 cases identified in 2009. The only way humans can contract the disease is through being bitten by an infected mosquito. Its incubation period is typically 7 to 9 days, but can be up to 21 days for some. The condition is very similar to another infection that is also found in Australia, called the Ross River virus. Given both viruses are very similar, the main difference between them is the Barmah forest virus is shorter in length and less severe. The Center for Disease Control reports that if you get the infection once, it is probable that your immune system will build up resistance against it. There is a 100% chance of recovery from the virus, and it is not fatal. BFV is a notifiable disease under the Health Act. This means a doctor who diagnoses the virus in a patient is required to inform the Department of Health so that more can be learned about its distribution and how to further control and prevent it. The combination of symptoms that characterize the Barmah forest virus include a fever, chills, fatigue, swollen lymph nodes, dizziness, lightheadedness, headache, lethargy, skin rash, joint pain, and tenderness of the muscles and tendons. The most commonly affected joints are the ankles, fingers, knees, elbows, shoulders, jaw, and wrists, with a rash typically developing on the limbs and trunk of the body. Some symptoms are more dominant or lacking, as the disease's effects vary between infected individuals. Less common symptoms include sore eyes, a sore throat, nausea, and tingling in the palms of the hands and soles of the feet. The primary symptoms previously listed sometimes last for up to half a year or longer in minority of cases. Those who suffer the disease for a longer period of time are at risk for becoming clinically depressed due to the emotional distress it can cause. Although the feeling of malaise fades within a few days, there are other primary symptoms that linger. Fever, nausea, and skin rash usually disappear within the first 2 weeks of the illness. However, joint, muscle, and tendon pain lasts much longer, causing severe distress. Some adults recover within 2 to 6 weeks, but many are unwell for at least 3 months. Unfortunately, symptoms may recur suddenly and without warning. Since 1992, there have been up to 700 cases of Barmah forest virus infections reported annually. It was in 1992 that the first epidemic due to BFV occurred in Northern Territory, Australia. This virus currently accounts for 10% of cases of epidemic polyarthritis-like illness in Australia. For this reason, the life cycles of mosquitoes and black midges are gaining attention worldwide because they are efficient vectors for multiple viruses. These viruses include the Barmah forest virus, Ross River virus, and canine heartworm, all of which are found in Australia. The BFV was discovered to be transmitted by mosquitoes in 1974 in the Barmah forest, of course, in northern Victoria, Australia. Although this was where the virus was originally isolated, it was not known that it could cause disease in humans until 1986. A patient who was demonstrating polyepidemic arthritis was thoroughly examined, leading to the isolation of BFV in humans. The Barmah forest virus is only found in Australia. Although the disease is typically found in eastern states, such as Brisbane, Queensland, and Victoria, it is beginning to emerge in Western Australia and Northern Territory. The main risk season to become infected with BFV is from December to May, with emphasis on the month of February, when the population of mosquitoes is highest. Current theories that exist for the prevalence of the BFV infection in Australia include its growing human population and also its respective climate and landscape. In particular, the heavy rainfall, large bodies of water, and their high tides provide suitable habitats for mosquitoes to prevail. These conditions allow the mosquitoes to live long enough to be able to acquire the virus and further pass it on to humans. Trewin and colleagues reported that in the early 2000s, extended drought conditions in this area resulted in rainwater harvesting. This unnatural increase in water storage in residential areas led to more niches for mosquitoes to exploit to reproduce and lay their eggs. In 2009, Barton and Weaver found a correlation between heavy rainfall, the increase in the population density of mosquitoes, and the activity of the mosquito-borne diseases. Flooding has also been associated with viral outbreaks as well. The Barmah forest virus is used for both the common and scientific name. It is an alpha virus of the family Togaviridae that is 60 to 70 nanometers wide. Similar to all alpha viruses, it is small and spherical. It has a positive single-stranded RNA genome of 11,488 nucleotides. To date, no studies have been carried out to investigate and identify the pathogenesis of the BFV disease. It seems likely that the virus evolved in the Old World from an insect-borne plant virus. The BFV infection cannot be spread from person to person. It is transmitted to humans by the bite of an infected mosquito. Mosquitoes are a part of the family Calicidae, which is in the order Diptera of the class Hexapoda, also known as Insecta, of the arthropod phylum. Various species of mosquitoes are efficient arbovirus vectors for this disease. Specifically, Calyx onolirostris, common bandit mosquito, Oshirlotatus vigilax, salt marsh mosquito, Oshirlotatus camptorhinus, southern salt marsh mosquito, and Colicoides marksii are carriers of the infection. This virus can only be transmitted by the bite of female mosquitoes requiring blood as a source of nutrition to lay their eggs. Fortunately, many go unharmed when bitten by an infected mosquito, especially children. Infections tend to be common in older people between the ages of 35 and 65. In addition to humans being vulnerable to this vector, marsupials, kangaroos, and possums, which are native animals of Australia, are commonly infected. The life cycle of the virus that causes BFV disease involves mosquitoes and vertebrate animals. An adult female mosquito that bites an infected animal, such as a bird or mammal, will pick up the virus. Please note, mosquitoes can also acquire the virus by biting a human that has been infected only if the bite occurs within the first 2 weeks after the birth of the infection. Then, the virus will multiply in the salivary glands of the infected mosquito. Once this infected mosquito bites another animal, the BFV disease is passed on, and the new host becomes infected. The virus multiplies inside the body cells, forcing them to manufacture more viruses, which burst out and go on to infecting other cells of the host. The BFV infection is diagnosed by a blood test that measures 2 classes of antibodies, IgG and IgM immunoglobulins. Diagnosis can only be considered conclusive if there is a 4-fold rise in the IgG antibodies. This must be detected in at least 2 blood samples taken 2 weeks apart from each other, with the first one from the beginning of the infection. This is determined by performing a serial dilution of sera using a hemagglutination inhibition assay. It is important to note that the IgM antibodies can be present in the body for a long period of time and is therefore an unreliable diagnostic test, and it has a high false positive rate. The ELISA assay is also useful as a diagnostic test and can be performed to detect the presence of IgM and IgG antibodies. There is no vaccine to prevent the infection, and there is no medical cure for the disease. Although there is no specific treatment for the virus, it is not fatal, and most individuals infected will recover. Medical treatment is aimed at easing joint pain and swelling, and minimizing fatigue by resting, drinking lots of fluids, mildly exercising, and eating healthy food items that are low in fat and cholesterol. For some individuals, simple painkillers such as aspirin is sufficient to ease the inflammation. It is important to focus on alleviating stress, physical fatigue, and substance use, as they may cause symptoms to worsen or to last longer. You are probably asking yourself, how can you prevent getting this disease? Well, you simply have to avoid getting bitten by a mosquito. Unfortunately, this isn't a simple task. Here are some suggestions that may reduce your chances of being bitten. Reduce the places on your property where mosquitoes can breed. Stay indoors when mosquitoes are most active, which is approximately 2 hours before sunset. Purchase fly screens, net hats, insect repellent that has DEET, keeping in mind that the repellent only provides protection for up to 4 hours. Avoid using scents such as perfume and deodorant. Wear protective clothing that covers the skin and is loosely fitted and light in color. Make use of electric insecticides. Place mosquito coils that are candle heated or gas operated devices using insecticide treated pads on your patio. Although vector control is complicated given the range mosquitoes can travel up to 5 kilometers, other general control measures can be taken to eliminate mosquito breeding sites. Dispose of all containers that hold water. Empty pot plant drip trays once a week or put sand around pot bases. Empty bird baths and pets' drinking water at least every 3 days. Boats and canoes should be overturned or have the drain plug removed. Screen all openings to tanks, wells, and other large water containers with wire gauze no coarser than a millimeter mesh. Keep roof gutters in good repair and regularly remove leaves and debris so that pools of water do not form. Dispose of all wastewater in such a way that no ponding occurs. Keep fish ponds, ornamental ponds, dams, and unused swimming pools stocked with fish which will eat the mosquito larvae. Keep swimming pools disinfected. Thank you so much for listening to my video on the Barmah forest virus. If you have any questions or concerns and want to further investigate information on this topic, you can check out the Centers for Disease Control and Prevention website or search any of the listed literature I have posted.